Okay, so welcome to topic 1.3. This is our intro to biological macromolecules. So macromolecules, of course, are these big molecules that are very important. We've been talking about them: proteins, nucleic acids, carbohydrates, and lipids. We talked a little bit about those. We're going to continue here talking about some specifics about some of these. First thing I want to talk about is something called functional groups, which is simply a group of covalently bonded atoms. Bonded atoms that have special properties. Now, these functional groups that we're going to talk about, this is typically where the action happens on these biological macromolecules. That's where something is going to occur to either change the structure, to add another monomer to some of these. It's going to occur at these functional group areas. So you're typically going to see, when I'm talking about just the functional groups, you'll see this letter R. Typically, this is just a variable, which means stuff is coming off there. Something is coming off there. The functional group itself is indicated right here. This is a hydroxyl group. Anytime you see an OH, that is a hydroxyl group. Next one, carboxyl group. Again, there's many more functional groups in these. These are just the four that I want to talk about because they're highly relevant to what we are going to be talking about. COOH. This is a carboxyl functional group over here. Again, this R indicates that you have something coming off of this carbon. No telling what. But yes, this is a carboxyl group, COOH, and I'll write the little chemical formula out here. COOH. Okay, the amino functional group. Here's the R again. Here's the functional group is NH2. I just see that it's NH3+, but the main thing to notice is that it has a nitrogen there. The amino group has a nitrogen. You can have an N over there. Okay, so let's move on to the next one. Phosphate, as you might guess, has a phosphorus inside, which is bonded to 4 different oxygens, right? 4 different oxygens. You'll probably see this a lot of the time without, let's look at the whole phosphate group here. But you'll see this a lot of the time, maybe missing 1 or 2 of these hydrogens. Big deal. Typically, phosphate groups can look something like that. PO4 might have a hydrogen sticking in here somewhere. But these are our 4 main functional groups. They're very important. It's important to memorize what these look like. If you would, pause the video real quick and look at these macromolecules over here on the right. And first, try and tell me what they are based on the atoms that you see involved. For instance, this one has nitrogen, oxygen, carbon, hydrogen. This one has phosphorus, oxygen, carbon, nitrogen. Tell me what you think they are, and then I want you to circle the functional groups and label them. Okay, you should have circled, and we know that at each of these junctures there's a carbon. You should have circled this here. What is that? That's a carboxyl. Carboxyl functional group. Of course, carboxyl has a hydroxyl group on there. I'll circle that as well. You have an amino group down here, right? This is another amino group, actually, even though it's bonded to a hydrogen, it has that nitrogen. But we'll just worry about this right now. This is an amino group. And of course, this right here is an amino acid. An amino acid. We'll talk about more in depth later. As an amino group, it has a carboxylic acid on the other side. Excuse me. All right, the second one. This is a nucleotide. Nucleotide. You should guess that this is a nucleic acid. This is a protein up here. So what functional groups do we see in here? We see this phosphate right over here, right? It's a phosphate group. We see a hydroxyl down here. See another amino group up here. So typically when you see these groups, that's where you know something is going to happen. Some sort of bonding is going to occur. Some sort of interaction with other biological macromolecules is going to occur. Okay. So first thing I want to talk about, we talk about biological macromolecules, is the concept of dehydration synthesis. So if you look up here, you have glucose, fructose, probably some things you've heard of. Well, what we have typically is we have an enzyme, which again, we'll talk about later, that kind of catalyzes these types of reactions. What that enzyme does is it pulls a water from this area, and again, what is this called again? This is a hydroxyl. hydroxyl functional group. And over here, another hydroxyl functional group. And basically what it does is it pulls this water molecule, these atoms that form a water molecule, and pulls it out of this molecule. And by doing that, it creates a bond. It creates a bond right here. In this case, this glycosidic linkage. So that is what is occurring in dehydration synthesis. It is the formation of a bond, I'll put between two monomers formed by removing H2O. Okay, so that is dehydration synthesis. Should clue you into that. Dehydrate, if you dehydrate something, you're removing water. And synthesis means to make, to synthesize something. Okay, hydrolysis. This is the reverse of that process. As you'll notice in this case, before we had glucose and fructose coming together to form sucrose, hydrolysis is the cleavage of a bond, or the breaking of a bond by adding water. Okay, so in this formula here, you'll notice you have oxygen, and in this case, you have the water entering and it's being split and kind of being added to each side of this, forming two monomers, right? So you started with sucrose, you've broken that down, and now you have glucose and fructose. That process is called hydrolysis. It might make sense to you because you have this word hydro, which kind of indicates water, possibly. Lysis means to break. Or I think it means to unform in Greek, right? So this is the process of hydrolysis. Make sure you know the difference between hydrolysis and dehydration synthesis. I'll leave it up here. You can see, again, in review, you have the removal of water. This is both of these processes on one screen, so you can kind of compare them. The removal of water to form a bond happening here. This is dehydration synthesis. I do recommend that you draw, I know this is kind of tedious, but maybe draw this happening right here so you can see. And hydrolysis. Bring over here. You have the addition of water, right? And it is breaking down this polymer into two monomers. Okay, that's it for topic 1.3. Thank you for listening.